Okay, good morning, everyone. Just a couple of announcements at the start. As a couple of you noticed, there was a typo in one of the slides on the last lecture. This term here shouldn't have been a 1 plus VA. It should have been VA minus 1. I've got that right. So I will update the notes and correct that. Thanks for those who spotted that. If I do make mistakes or you think something doesn't look right, please do feel free to correct me. Usually I say to people, I'm not actually thinking that much about the math itself when I'm lecturing. I'm thinking about, am I speaking clearly? Am I, if people paying attention, are people having confused looks and things like that? And so I'm not usually solving things up here. So sometimes, yeah, if I make a mistake, it's because I'm not really thinking about the math and I've maybe just made a typo in my notes or something like that. So please feel free to interrupt. The other thing, a couple of people asked about the biology and were a little bit concerned about a lot of the terminology that was introduced. I would say that this lecture and the lecture from last week were particularly heavy in terms of terminology. As the course progresses, it's not going to be that biology heavy every single lecture. This one is, again, there are quite a few terms to introduce. So kind of bear with it a little bit. It's not going to be like this for the entire course. Inevitably at the start, there's a certain amount of biology that you need to be taught, and then we'll be kind of reusing those concepts over and over again. That being said, I've uploaded a glossary of biological terms onto the course schedule materials page, and I'll update that throughout the course as well. And also, during the lectures or afterwards, I've got office hours as well today after this lecture. If you have any questions about the math or about the biology and you want me to just clarify them, please feel free to interrupt me. Okay, so there's no problem. I'm always happy to do that. And I would also say the extent to which you need to understand a lot of that biology terminology is relatively shallow. So, for example, we talked a lot about alleles the other day. An allele is just a variant of a gene. Okay, so there are different people have different alleles. That's kind of the extent to which you know it. Obviously, you can, look in textbooks or online resources on like YouTube or Wikipedia if you want to learn about alleles in a lot more detail. But the extent to which you need to know about them is they are just variants of different genes as far as this is concerned. Similar for many of the other concepts that we'll talk about. Okay, so today we're going to be talking about diploid models. The other day we talked about haploid models. So today we're going to be thinking about extending some of those ideas into this slightly different genetic structure. Why do we care about diploids? Well, lots and lots of things are diploids, including us in this room. Many plants, animals are diploid. It's kind of one of the main modes of translating genetic material from generations is going from a diploid phase into a haploid phase. So that's why we also care about that haploid phase as well. So we need to be thinking about models of selection in the context of diploids. Like I said, there are quite a lot of biology background or terminology to understand in the context of these diploid models as well. Like I said, as the semester goes on, this list of concepts will gradually shrink as we go into future lectures. But like I said, if there's anything that you want me to explain in more detail, please feel free to interrupt. Okay, so ploidy. What is ploidy? Well, ploidy is essentially just how many chromosomes a typical cell has. And so when we talk about individual cells, or we could be talking about organisms, we refer to organisms having diploid or haploid cells, but we also refer to organisms themselves as being diploid or haploid, or possibly triploid or tetraploid. So think of capital N as just the number of chromosomes that an individual cell has, then haploid has N, diploid 2N, and so on. So diploids are interesting because they have one copy of each chromosome from each parent. Okay, so our sex cells that we produce over in sperm are haploid, but when they form a new zygote, a new individual, they end up becoming diploid. So you get one, these inherited from each parent. We're not going to be thinking about triploids and tetraploids. They're just there to let you know that there are other things. This is more common in things like plants as well. So we're not going to be thinking about anything above diploids in this course. So we want to think about evolution in diploids because they're so common, because we're diploid. But how do we go about modeling them? Well, we can use some of the tools we learned from haploids because haploids are relatively easy to model. Okay, so Haploids are easy because they don't have this process of segregation and fertilization that we have. So segregation is just that translation of your normal diploid cells with 2 copies of each chromosome, and then splitting down into haploid cells, so creating things like sperms and eggs. And fertilization is just the reverse of that process when a sperm and an egg fuse together. I'm using sperm and egg, but that's more generally gametes fuse together to form a new individual. So you go from those haploid cells into a diploid cell. Okay, so you go from N to 2N at that stage. Haploids are nice because in the case where we just say have 2 alleles, big A and little a, we only had to keep track of those 2 things, those allele frequencies, and it turns out the allele frequencies and genotypes are the same thing when we're talking about haploids with 1 locus. Now we have to keep, even in this very simple case, track of at least 3 genotypes. So individuals that have 2 big A's, 1 big A, 1 little a, and 2 little a's. Now, I said 3 here. There are cases where it might matter which parent you inherit those alleles from. So it's at least 3, it could be 4, but we're going to really be focusing on the case where it doesn't matter which one you've inherited them from. A couple of bits of terminology here. Homozygotes, homo meaning the same, are individuals or cells that have identical alleles on each of their chromosomes. Now, when I say this as well, this is the chromosome, like the alleles that we actually care about. They might not be homozygous everywhere in their genomes, but the ones that we care about, this particular genetic locus, they're homozygous. They have the same alleles. And if they're not homozygous, if they have different alleles, then they're heterozygous, hetero meaning different. Okay? So homozygous, they have the same alleles. Heterozygous, they have different alleles. So we could follow a similar approach as we did last week with our haploid models, and we could track those genotypes. Like I said, that's a little bit more complicated. Obviously, we're always going to have at least as many genotypes. Typically, we'll have more genotypes than alleles, right? Because our alleles are forming the different types of genotypes in different combinations. So it's much easier if we can track things at the gamete stage, because think about the life cycle of like a human. We'll go through most of our lives as diploids, but then when sex occurs, we're splitting up things up into gametes. So the reproduction stage, we have to kind of get filtered through that gamete stage. So why not census the population there rather than doing it at the more complicated stage? That's why we're going to think about haploids again, but thinking about how we're forming our zygotes, and therefore ultimately that leads us to our diploid models, but in a slightly simpler way. Okay, so one thing we're going to be assuming, I've mentioned this word zygotes a few times. Zygotes, think about as just sort of your newborn cells, your newborn individuals that are formed from your gametes. So we're going to assume that there's no preferential mating. So mating is random. That means the population is, things are well mixed. There's no preference going on here. And a big A is not more likely to associate with another big A, for example. And we're going to be thinking about our genotype frequencies from our allele frequencies. Okay. So let's start by thinking about a really simple scenario. We're thinking about the dynamics within a single generation. So let's not think about change over time yet, just one generation. And we're going to let P and Q be the frequencies of our big A and little a gametes this time, respectively. So what is the probability, given that we're going to have random mating, what is the probability of forming each genotype from a large pool of gametes under random mating? Any idea here? So what we want to have ultimately is we want to have, what's the probability of, say, big A, probability of big A, little a, and the probability of little a, little a. Given that everyone's moving around, mating randomly, it's a large population, so that's basically saying we don't have to worry about random effects. Everything basically behaves deterministically in a large enough population. We got frequency of big A being P and little a being Q. So what's the probability that 2 big A alleles, big A gametes, are going to come together to form a big A genotype? Think of this as taking balls from a jar. Okay, yeah.
3%.
Sorry.
3%.
1/3. So not 1/3 in this case. Think about, okay, so suppose we've got a jar. It's got these lots and lots of colored balls in it. One of those colors is blue, one of those colors is red. Okay, what's the probability if, say, suppose the frequency of the blue ball is P. What's my probability of pulling a blue ball out first? That'll be if I pull two out, so you've actually jumped ahead, but that's good. Yeah, so you pull out one first, it's P, and then because it's large, we're assuming that there's no sort of effects on sort of the probability of pulling out another one. Pulling out another one is going to be P again in a sufficiently large population. So P times by P, so we have P squared. So here, P times by P is P squared. Okay, by symmetry, what about the bottom one? The probability of pulling out two red balls or two little a alleles?
Q squared.
Q squared, exactly, yeah. U times by Q is Q squared. Okay, slightly more complicated for our heterozygote. What about the probability of a big A gamete and a little a gamete? 2pq. Why 2pq?
It's like big A, small a, and small a, big A.
Exactly, well done. So we could pull out a big A first and then a little a, or we could pull out a little a and then a big A. So we end up with 2pq. Yes.
Well, one of the earlier slides you talked about three genotypes: big A, big A, big A, little A, little A, little A. But there was no little A and big A. Why is that?
That's because, so you can think of this one here as being big A, and this one here as being little a, big A. So I said earlier that we're going to assume that the parent that you inherit them from. So suppose this is the mother and this is the father. It doesn't matter. We're assuming the which one you inherit them from. So something like your eye color, for example, doesn't matter which allele comes from which parent. There are some cases that are like sex-linked traits that do matter, but we're going to just assume for simplicity that it doesn't matter. Therefore, yeah, that's the big A, little a is the same as little a, big A. The order doesn't matter. Okay. Any other questions? Okay. So we can write that information in something called a Punnett square. This is named after a British geneticist. So a Punnett square is just a different way of writing down that information. Sometimes can help you think about how we calculate those things. Okay, so if we set up a square that looks like this, we're going to have, for example, our frequency of, say, female gametes here. And we have big A and little a as our possibilities. And likewise, we're going to have a frequency of male gametes. And then, like we've just done, we can think about what is the frequency of each of these different gametes. And therefore, we just multiply the frequencies together to get each of these different boxes. So big A, we have a P times by P, so that's where we get our P squared from. Likewise, little a, we have Q times by Q, so we get Q squared here. And then big A, little a, going to be PQ. Little a, big A. All right, it's QP, just so it's clear the order here. Obviously, these two things, PQ and QP, are the same. So that's why we end up with our 2PQ. This is a very famous result, very simple result, but these are known as the Hardy-Weinberg frequencies. It's named after a couple of theoretical biologists early in the 20th century. So Hardy-Weinberg frequencies are basically this very simple null hypothesis about the frequencies that alleles should be in a population if there's no other effects going on. Okay. Should be pretty obvious as well that if we sum these things together, P squared plus 2PQ plus Q squared, we can factor this into P plus Q all squared, because P and Q are just the frequencies of our alleles. Q is equal to 1 minus P. So this is just equal to 1. Okay. So everything sums to 1. So like I said, this is kind of like a null scenario. Nothing's really happening. These are the frequencies before selection, therefore. If there was no selection, things ultimately don't change. We'll talk about that in a few slides. But if we do have selection going on, then we need to think about what happens to these frequencies as we introduce variation in fitness between our different genotypes and hence our different gametes. Okay. So one thing we can say is that since selection alters the frequency of each genotype proportional to its fitness, that's kind of by definition what selection is doing. It's going to be, things that are above average fitness are going to be increasing in frequency. Things that are below average fitness are going to be decreasing in frequency. That's all fitness is all about, essentially. We say the mean or absolute fitness of the population. Sorry, this is the mean absolute fitness. Not mean or absolute. The mean absolute fitness of the population, W bar, is just going to be a weighted sum over those different frequencies. So we've got the frequencies up here of our different genotypes that are coming about by random mating. And then if we define W, ij, as we had in the last time, this time it's going to be for the genotype ij, to be the fitness of that genotype, then we start off with our p squared plus 2pq plus q squared as our baseline frequencies of those different genotypes. That's just from what we had from the previous slide. We now have a weighted sum. So we just multiply the frequency of each of those genotypes by the fitness of those genotypes. So Wij for each of those combinations. We haven't talked about exactly how we're defining these Wijs. That doesn't matter at this stage. All we're doing is we're saying the average or mean fitness of the population here is a weighted sum of the different fitnesses and their frequencies. Okay, and because we're saying that the frequency of each genotype is changing proportional to its fitness, we can work out the frequencies of those different genotypes after selection. Okay, so for example, our big A genotype, well, it was at frequency p squared before selection. Now it's going to be changing according to its relative fitness. What's its relative fitness? Well, its absolute fitness is W sub capital A, capital A. And because we're thinking about relative fitness, we divide by its mean, the mean fitness of the population. Hopefully, intuitively, this should make some sense. We start off at a frequency of p squared. And then if WAA, the fitness of this genotype, is above average, then it's going to increase in frequency. If it's below average, if it's below W bar, then it's going to decrease in frequency. We do the same for our other genotypes. For our heterozygote, we're going to have the fitness of the heterozygote times by its frequency 2pq divided by the mean fitness of the population. And finally, for our other homozygote, again, its fitness times by q squared, its frequency before selection, divided by the mean fitness of the population. Any questions? Okay, the final step after selection is going to be the production of gametes. Now, we're going to be ignoring mutation. Remember, this is a process we mentioned earlier. Mutation can change the alleles. We're going to be dealing with that in a later lecture. We're not going to be talking about mutation here. So basically, that just means that things that are inherited perfectly. It's perfect copies. So now we can think about the proportion of gametes that are produced from each genotype. So remember, what we've done is we've gone from, okay, we've got our gametes. What are the frequencies through random mating that give us our different genotypes? Then we're going to be adjusting those frequencies according to their relative fitnesses. And then we need to produce gametes again. So we've gone from the gametes at the haploid stage to our diploids, and then we're completing that life cycle by producing gametes again. So let's think about our different genotypes. We've got 3 different types, remember. Our homozygotes, big A, and little a, and then our heterozygotes, big A, little a. And then we can think about the proportion of gametes. And we've got 2 types of gametes, big A and little a. Again, putting stuff in tables often helps. Make sure that you don't miss anything out. Okay, so what proportion of big A gametes do my big A homozygotes produce? Somewhere between 0 and 100%. It is. So big A individuals, they need to produce some gametes. They're either going to produce big A gametes or little a gametes or some combination of there. Yeah, what are they going to produce? Exactly. Yeah. So 100% or 1, a proportion of 1 of their gametes are going to be big A. There's no mutation here. So if, for example, we had a mutation rate of, say, let's say mu, we would have something like this. They're producing some individuals of the different allele because there are mutations. We don't have this in this particular model. We have this perfect inheritance. Okay, what about little a? What proportion of their gametes are big A? 0. And then little a has to be 1, right? Because they have to sum to 1. And then the middle, the heterozygotes. What goes in each of these boxes altogether? 0.5, 0.5, yeah. So half of each. One way of thinking about it, if this is slightly less clear, is, I'm just going to zoom in here. You can think of them as having, say, a split coming down here. Okay, so they produce equal, there's no bias in what they're producing, right? So half of the things they produce are going to be A, and half of the things they produce are going to be little a. If it was big A, then again, we have a half of them are A, big A, and half of them are big A as well. That's all that's happening here. So that's all the background, the setup. There's quite a lot of setup going into this. We need to think about that haploid stage where there are gametes, the frequency of the different genotypes coming from those gametes, so the zygotes that are formed. We then need to think about selection, so how it's differentially affecting the survival of those individuals, and then ultimately the reproduction again at the gamete stage. If you recall, this was all in one generation, right? So you've just done that life cycle going from gamete, diploid, back to gamete again in one life cycle. What we really care about, though, is what's happening between generations over a long period of time, right? So we want to replace our P's and Q's that are just within a generation to time-dependent P's and Q's. So thinking about what happens over multiple generations. And we want to derive the frequency of these different gametes in the next generation based on what we have in the current generation. So we need to account for these things like selection and the production of gametes when we're doing so. So what we're going to do is we're going to adapt what we had before into a time-dependent equation. Okay, so our frequency of big A allele is going to be P, and we're thinking about it in the next generation. So we're in generation T, and we're thinking about what's going on in generation T plus 1. What's that going to be? Well, again, we're thinking about essentially weighting some sums. We need to think about the production of each type of gamete after selection. So essentially, we're going to combine our information on this slide with our information down here at the bottom on this slide. So we know, for example, change my color, that for our big A genotype, it has this frequency after selection. Its fitness is relative fitness multiplied by its frequency before selection. So we'll start with that. So we have W sub big A over B bar. And we also had up here its frequency. So it was a frequency of P of T squared this time, because we changed everything to time dependent in this particular generation. So all I've done there is taken what was on what I highlighted in pink and made it time dependent. And we need to multiply this by the proportion of gametes that are produced by those individuals of that genotype that are of big A allele. Okay, so here it's going to be times by 1. So obviously it's just going to be, what was that before? Okay, so remember this is the frequency of our big A allele in the next generation. And this term here is telling me the frequency of our big A allele arising from individuals who end up as big A, homozygotes. That's not all though, because we need to think about our heterozygotes, because those individuals have one copy of our big A allele. So we need to think about their contribution to the next generation as well. But all we need to do is exactly what we did before. So change my highlight now. Let's start off with our fitness. So this is the fitness. Sorry, this is the fitness adjusted frequency. So after selection of heterozygotes. So we just copy that down here. So we had the relative fitness of those individuals times by 2 P of T, Q of T. And that's all I've done there is I've just taken what I had on that previous slide. I made it time dependent. Made any mistakes? Good. And then we need to, what do we need to do to this? Yep. Multiply by half. Why by half?
We in our table before we decided that 1/2 of them produce the big A.
Exactly. So we're multiplying by this box here. A half. The nice thing here is the half and the 2 are going to cancel. So we can write this now also over one big fraction. So we can have, clear that half and that 2 are going to cancel. We have a common P of T that we can pull out the front. Likewise, we have a W bar. They're both over the same denominator. So let's pull this P of T out the front. The mean fitness appears on the bottom and is the only thing that appears on the bottom. And then what have we got? Well, we're still going to have a P of T times by our fitness of our homozygote. And then we're going to have the fitness of our heterozygote times by Q of T. This term here. And remember, Q of T is just 1 minus P of T, because these are both frequencies, right? So we can eliminate the Q of T by writing this as 1 minus P of T. So now we have an equation that tells us, based on the current frequencies of the A, or the big A and little a gametes, it's the big A and little a alleles, in this population in the current generation, and their relative fitnesses, what's going to be their frequency in the next generation? And this is known as the Fisher-Haldane-Wright equation of population genetics. This is kind of a big equation. Yes.
So there were 2 gametes in the beginning, frequencies P and Q, and there was a fertilization, there's a zygote, and then there's segregation, and we have 2 gametes again, and we want to figure out the frequencies of new gametes, right? Based on the frequencies that we had before and those selection parameters.
Exactly, yeah. And yeah, because we're assuming, I mean, yeah, those dynamics you just described occur within a single generation. And if we just care about a single generation, then we can say kind of on average, what is the A and big A and little a alleles? So gametes alleles kind of blur together at that point. So this equation is nice. It's just a difference equation. We had a difference equation before with our haploid population. This is a little bit more complicated, right? Because we've now got these heterozygotes to account for, and we have to think about this segregation and formation of gametes. Ultimately, it is just a difference equation. It's it's going to be dependent on, this W bar down here, just remember that this is going to depend on the frequencies of P and Q as well, right? So although it's not written as a function of P, that is also a function of P. It's not just a constant. But ultimately, so we've got some nonlinear difference equation that is going to propagate forwards. If we know what the frequencies and the fitnesses are in each generation, then we can propagate things forwards. These three here, Fisher, Haldane, and Wright, were sort of three big dogs in evolutionary biology in the early 20th century that kind of helped do a lot of the mathematical formalization of ideas that had just kind of been floating a bit around in biology before. So they really tried to like pin down what are the key mathematical principles, like can we make predictions or understand what's going on with selection. So this is a very famous equation. There is another way of writing down this equation, which I'm not going to show you the derivation of because it's kind of involved. I'm actually going to leave it as an exercise to prove that these two things are equivalent, but we can write that in a slightly different format. And this format can be useful for certain reasons. I'll explain why, or it'll become clear in a moment. So the key thing here is that we have a P of T out front. So that means that we can think relatively easily about delta P, so the change from one generation to the next, because we can just subtract P of T from both sides. Another nice thing about this is these terms here tell us something about the relationship in fitness between our homozygotes and our heterozygotes. So if you think from the perspective of these alleles, they're spending some of their time on average in homozygotes, so with an identical copy of themselves, and some of their time is being spent with the other allele that they're potentially competing against in the population, right? So there's a little bit of a potential conflict here. Right? But ultimately, these two terms can tell us something fundamental about when we would expect there to be variation in the population maintained. So that's what we'll get onto now.
Question about
Yes.
the previous slide.
This one here.
The frequency of the big and small gametes, right? And
Yeah.
we looked at like the mean fitness,
Mhm.
the actual fitness of like the homozygotes and the heterozygotes, but what about like the small a's?
So small a's will be done by exactly the same process. Obviously, because P and Q here will both always sum to 1, as soon as we know P, we can know Q. So you can work out the frequency in the next generation of the little a allele just by doing 1 minus this. So you could do a check of doing 1 minus this and also calculating from first principles like we've done here, what is the corresponding equation for Q would be. Arbitrarily, we've picked the big A allele and P. We could have done exactly the same and phrased things in terms of little a allele and Q. But yeah, I'll leave that as an exercise as well, because it's just essentially algebraic manipulation there. 1 minus this, and also going from first principles will give you ultimately the same thing. Any other questions? Okay. Time for an exercise for you guys. See how you guys get on with this. So one more term here that I want to introduce is this idea of polymorphism. Sounds a little bit scary, but polymorphism is basically just, you can think of splitting this into two words: poly, meaning many or more than one, and morphism, more different types. Okay, so polymorphism, just more than one type in the population. That's all it means. So we say a stable polymorphism exists when at least 2 alleles coexist at the same locus. So they're coexisting and they're stably coexisting. So why does that matter? Well, what we can use the Fisher-Haldane-Wright equation, and in particular, we'll use this format here. Hopefully I can get both of these things on the same slide. Can we learn anything about when stable polymorphism is going to occur based on this equation alone? Now we could, do stability analysis on this model, and you can do that totally to find out, where are your steady states? when are they stable, when are they unstable, depending on things here. But we can also work something out very straightforward just by looking at this equation. So I'm going to give you a little bit of a hint, but I want you guys to think about how we might work out when we get stable polymorphism. What does this mean mathematically, stable polymorphism? So for there to be a stable polymorphism, that means that we need some P star Q star. So we need P star Q star with what? Well, P star and Q star, they're just values between 0 and 1. But for us to be a stable polymorphism, by definition, we need these to be between 0 and 1, but not equal to 0 or 1. Remember, these are just frequencies, right? So if P is at frequency 0, Q is at frequency 1. Likewise, if Q is at frequency 0, P is at frequency 1. So if either of these is at the frequency of 0 or 1, that by definition means that there's only one allele in the population. So we need them to be, we need to find a steady state, essentially, where both P and Q are non-zero. So that's what you can do in terms of, from a mathematical perspective. You can do that full stability analysis, but can we just find what that steady state would be by looking at the terms in this equation here? And so the hint I'll give you is think about delta P being equal to 0. Why? Well, if delta P is equal to 0, then that means there's no change between generations. So when do we get delta P is equal to 0? Based on this equation above. Remember, your delta P is just equal to P of t plus 1 minus P of t. So have a think about that, discuss with your neighbor as well. I'll walk around and see what you guys think. So let me put both of those things on the screen so that's still visible. Okay. See how you guys get on with that. Just to be clear as well, I'm not asking you to solve the equation. All I want you to do is essentially look at that equation and make an inference based on the terms that are in that equation. In order for delta P to be equal to 0, what must we have? I'm thinking especially about our fitnesses. Those two things that are highlighted. Okay, so if the fitnesses are all the same, then everything kind of exists because nothing interesting is happening. I should say, yeah, we're assuming there are some fitness differences, but that is a totally valid answer to what I posed here. Let me go through this with you guys, see what you think. So I've heard a couple of people, kind of a few murmurings along the right lines. So if delta, sorry?
I saw P.
Sure, go for it. See, tell me what you think. Like, what's your biological interpretation? I don't want to hear your mathematical interpretation. I want to hear what's your biological interpretation.
I haven't thought about biological interpretation yet, but
okay.
Since we want frequencies for which neither P or no Q are 0, so for delta P to be 0, that's the other term in the numerator, like WAA minus WB. The whole thing should be equal to 0.
Yeah.
And we know that Q is 1 minus P.
Mm-hmm.
We can set that equal to 0 and solve for P.
Yeah.
But we haven't checked stability yet by doing that.
Sure.
And I haven't thought about biological interpretation right now. And we can
okay.
talk about it.
So yeah, so you're kind of like partway along there. We're not, yeah. Think of this as, yeah, we're not going to talk about the stability. We're just going to think about like what's kind of a prerequisite for this. So you mentioned that term that was in the numerator. So we have the difference in fitnesses here for our big A homozygote and heterozygote. And at equilibrium, that's going to be a frequency of P star on the outside. And then this is added to, similarly, our fitness for our heterozygote minus the fitness of our little a homozygote. That's by Q star. And this has to be equal to 0, right? For our delta P to be equal to 0. So this is just a number. This is just a number. AX plus BY is equal to 0. If X and Y are both positive, what does that tell you about A and B? Maybe I shouldn't have chosen A and B here. Let's choose C and D just to make sure it's clear. So if X and Y are both positive, what does it tell you about C and D? For this to be
Positive, one of the others.
one has to be positive, one has to be negative. So it tells you that the fitness here, this expression here, has to have a different sign to this expression here in order for us to have polymorphism. So what does that mean? That means that either our heterozygote has to be fitter than our homozygotes, or it has to be less fit. So these things have to have opposite signs. So the biological interpretation that I'm kind of seeking from there is that in order for us to have stable polymorphism, our heterozygotes have to either have higher fitness or lower fitness in order relative to our homozygotes in order to get this stable polymorphism. I won't write that all out, but it's in the notes. So again, this is something like quite fundamental that we can learn about how populations are structured genetically in order to get it from this relatively simple model. It's quite a profound result. Okay. We talked a little bit, we kind of glossed over what happens if they have the same fitness. Well, if they have the same fitness, what happens? Well, in that case, all of our fitnesses, all of our relative fitnesses are going to be equal to what? All of our genotypes have the same fitness. What's going to be the mean fitness of the population? If every individual has the same fitness, so suppose everyone in here, let's think of a good example. Suppose everyone in here had the same height, for example. You'd all have this, the average height would be identical for everyone, as well as being the same as their actual height, right? So it's the same here for fitness. So this is all just W bar over W bar in each of these cases, right? So it's all just equal to 1. What does that mean? Well, if we go all the way back up to our, well, we can do it from the Fisher-Haldane-Wright equation, or we can think about what happens from on this slide here. All of these relative fitnesses are just going to be equal to 1. So after selection, everything just stays in Hardy-Weinberg equilibrium that we started with, p squared, 2pq, and q squared, because everything cancels out. You can see that ultimately from here as well. So we can kind of show that if you want. So just to show that explicitly, if, for example, we set each of these frequencies equal to some value w, and that's for all ij in this set of alleles. I'm going to call this W bar just to make sure it's clear. Okay. Then what do we have from our Fisher-Haldane-Wright equation? Well, the frequency of P in the next generation was P of T times by, in this case, all of our fitnesses are going to be replaced with W bar. So we can have W bar P of T plus W bar times by 1 minus P of T all over W bar. These W bars are going to cancel out. And then we're going to have P of T times by, in brackets, P of T plus 1 minus P of T. So it's just going to be 1. So this is just going to be P of T. So if there is no variation in fitness, everything just stays in Hardy-Weinberg equilibrium. So again, this, I don't know how intuitive this is, but suppose you had a random any sort of setup, any initial values of P and Q, right? Any initial frequencies of big A and little a alleles in the population. And then after one generation of random mating, if there's no variation in fitness, then everything will just end up in Hardy-Weinberg equilibrium. So no deviation from Hardy-Weinberg equilibrium. So basically, this is all this is saying is like once things settle down into Hardy-Weinberg equilibrium, they stay there. So in some states, it's a steady state. Okay, we've got one more quick definition to do, and then we're done. So this is just a mathematical definition, a way of rewriting things as we often do in these models. Sometimes writing them in a slightly different format gives us a slightly different interpretation to things. So if we define W sub A, so just one of those alleles, to be the mean fitness of that allele, and this is just weighted by its frequency in homozygotes and heterozygotes. So if we define that to be our mean fitness of that allele, we can then rewrite our overall fitness, our mean fitness, in terms of the frequencies of those alleles and the fitness of those alleles. It's just a rewriting of what we had before. Again, I'm not going to show that. I'll leave that as an exercise if you want to derive that. It's just a algebraic manipulation to get from what we had before to getting these expressions here. But the interesting thing is that we can simplify our Fisher-Haldane-Wright equation, which before looked like this. W big AA times by P of T plus W big A little a. I'll write it as Q of T this time. Times by P of T all over mean fitness. Well, this term here on the top, the reason we chose this interpretation up here is this is the same as this, these two highlighted things, right? So I can just replace it. Since we have the mean fitness of that allele times by its frequency divided by the mean fitness of the population. Yeah, we haven't created any new information in here. All we've done is we've done a clever bit of substitution here, but it tells you again something about like the change in the frequency of this allele ultimately depends on the frequency of that allele and its fitness. And this should hopefully look very similar to what we had in our haploid model, right? It's basically the same thing. And very finally, we can then say the change in the frequency of an allele is just going to be the difference between generations. And so what did we have on our previous model here? Well, this was the frequency in the next generation. If we subtract P of T from this, then we're just going to have the fitness of that allele minus the mean fitness of the population times by P of T over W bar. Okay, so all I've done to this is I've just subtracted P of T and then combined them over the same fraction. And this is kind of nice as well because it's again just a rewriting of things. But this term here, W sub A minus W bar, is just referred to as the mean excess fitness. It's just again another way of writing things. Again, intuitively, if the allele is above average frequency, then this is going to, is above average fitness, then this is going to be positive. So delta P will be positive. If it is below average fitness, then it will be negative. Therefore, delta P is negative, and therefore it's going to decrease in frequency. Okay, so it tells us the relative amount that it's going to increase or decrease per generation. Okay, that is it for today. I say today we've talked a lot. Again, I appreciate there's a lot of terminology to take in. This will decrease over time. These diploid models are essentially just extending some of the ideas we had in haploid models that are obviously a bit more complicated. A question I'll leave this as a challenge as well is, I think I put this actually even on the problem sheet is, oh no, I haven't put this one on the problem sheet, I think, but we can derive this from first principles. So if you want to do that as an exercise, I'll leave that as an exercise. Other than that, I'll see you guys on Wednesday. Oh, I've got office hours now in K9525. I think that's my office number. So if anyone wants to ask something in more detail, that's fine.
Why in these models we are looking at the frequency of big A and little a? Like we put driver equation, writer equations to talk about frequencies of
The
those
genotypes?
different genotypes. And is it just because of simplicity? Because then we would have 3 different variables.
Exactly, yeah. And if you think this is the simplest case where we've just got 2 alleles, and there's also no difference between whether you're a big A, little a, or little a, big A, right? So no difference between where you inherited those alleles from. Very quickly, if you start having more alleles or more loci, your number of genotypes explodes, whereas your number of gametes stays relatively small. So it might not be super apparent, like, oh yeah, we can work with 3 or 4 equations. It's obviously easier to work with 2, but that number rapidly grows in terms of genotypes. Yeah. You guys are free to go.